Now, this is Introductory Molecular Biology and Diagnostics, our lectures 9 and 10. And in this, we'll be talking about general molecular techniques. Now, the objectives for this section, we'll be looking at DNA, RNA, and protein electrophoresis. Then we'll talk about utilization of molecular biology in our daily activities as laboratory personnel. So we're talking about DNA chips, automation, gene therapy. We'll also look at applications in diagnosis of genetic disorders. Then we'll consider the Human Genome Project, and then ethical considerations. Like electrophoresis, which you do every day in your lab, you can do same for DNA and RNA. So electrophoresis uses an electric field applied across a gel matrix to separate large molecules such as DNA, RNA, and proteins, and this depends on charge and size. Samples are loaded into the wells of a gel matrix that can separate molecules by size, and an electric field is applied across the gel. So just like you do in your labs, you apply electricity to it. This field causes negatively charged molecules to move towards the positive electrode. The gel matrix itself acts as a sieve through which the smallest molecules pass rapidly, while longer or larger molecules are slower moving. So smallest ones go through the matrix, which is acting as a sieve. The very large ones are slowed down in their movement. Now for DNA and RNA, sorting molecules by size in this manner is trivial because of the uniform negative charge on the phosphate backbone. So when it comes to charge, DNA and RNA have fixed or same charge. For proteins which vary in their charges, a clever trick must be employed to make them mimic nucleic acids. Now we're talking about polyacrylamide gel electrophoresis later. So for proteins, the charges vary. Now different kinds of gels have different pore sizes. Like sieves with finer or coarser meshes, some gels do a better job of separating smaller molecules, while others work better for larger ones. Gel electrophoresis may be used as a preparative technique, that is, when purifying proteins or nucleic acid, but most often it is used as an analytical tool. So the gel electrophoresis may be preparative or analytical. So let's pick agarose gel electrophoresis. So agarose gel electrophoresis is a technique used to separate nucleic acids primarily by size. Agarose is a polysaccharide obtained from seaweeds. It can be dissolved in boiling buffer and poured into a tray, where it sets up as it cools to form a slab. So when you pour it into a tray, it settles and cools to form a slab. It's like molten. Agarose gels are poured with a comb in place to make wells into which DNA or RNA samples are placed after the gel has solidified. The gel is immersed in a buffer and current is applied across the slab. So when you pour the agarose material or molten material into the tray, you fix a comb like the hair comb into it. Then it creates holes in them. These holes will be channels where you would place your preparation, your DNA, your RNA, your protein preparation for your electrophoresis to take place. So the double-stranded DNA has a uniform negative charge that occurs independent of the sequence of composition of the molecule. Therefore, if DNA fragments are placed in an electric field, they will migrate from the cathode towards the anode. The rate of migration is directly dependent on the ability of each DNA molecule to worm or wiggle its way through the sieving gel. the agarose matrix provides openings for macromolecules to move through. The largest macromolecules have the most difficult time navigating through the gel, whereas the smallest macromolecules slip through it the fastest. So you realize that those who are huge have longer kilo bits or sizes. They take a longer time to go through, while those with smaller kilo bases would take a shorter time to get through. But as always, for double-stranded DNA, the movement is from the cathode as a negative to the anode. Because they are negatively charged, they move towards the positive, which is the anode. Now, because electrophoresis uses an electric current as a force to drive the molecules through the matrix, the molecules being separated must be charged. Since the size to charge ratio for DNA and RNA is constant, for all sizes of these nucleic acids, the molecules simply sort on the basis of their size. The smallest move fastest and the largest move slowest. Like I said earlier, largest take time, smallest go quickly, they rush through. All fragments of a given size will migrate the same distance on the gel, forming the so-called bands on the gel. Visualization of the DNA fragments in the gel is made possible by the addition of a dye, such as ethidium bromide, which intercalates or fixes itself between the bases and fluoresces when viewed under ultraviolet light. So for visualizing the bands, you need to add a dye, that is ethidium bromide, and this causes a fluorescence that can be seen under ultraviolet light. Now, by running reference DNAs of known sizes alongside the samples, it is possible to determine the size of the DNA fragments in the sample. So just like the control you use for HB electrophoresis. So something should indicate to you which ones you have identified. It is useful to know that by convention, DNA fragments are not described by their molecular weights, unlike proteins, but their length in base pairs or kilo base pairs. So DNA fragments are measured in the length of their base pairs or kilo bases. Now I move to the next one. Polyacrylamide gel electrophoresis. Polyacrylamide gel electrophoresis page. Like DNA and RNA, proteins are large macromolecules. But unlike nucleic acids, proteins are not necessarily negatively charged. But nucleic acids are negatively charged because of the phosphate backbone. The charge on each protein depends on its unique amino acid sequence. Thus, the proteins in the mixture will not necessarily all move towards the anode. So if it becomes proteins, movement is not specifically to the anode. But for the nucleic acids, that's DNA, RNA, it is moved towards the anode. Additionally, whereas double-stranded DNA is rod-shaped, most proteins are globular. You know rod-shaped and globular? Yeah. Further, proteins are considerably smaller than nucleic acids. So the openings of the matrix of the agarose gel are simply too large to effectively provide separation. So that is another point. Consequently, unaltered proteins are not very good prospects for electrophoresis on agarose gels. To separate proteins by mass using electrophoresis, one must make several modifications. So if you want to use the page for proteins, you'd have to do modifications before it will work perfectly for your situation. So modifications need to be made to accommodate this. Now first, a matrix made by polymerizing and cross-linking acrylamide units is employed. So you think of cross-linking them. Now a monomeric acrylamide is polymerized. polymer, so bringing so many together. And the polymers are cross-linked using NN methylene bisacrylamide to create a mesh-like structure. So when you cross-link them, you also when you polymerize them, you also cross-link them to make it more into a mesh. So see it as a thread, then the thread is lined like in a zigzag manner, then the zigzags are connected to each other when they lie side by side, and see it as such. So in that case, it has been pushed into a mesh form. Now one can adjust the size of the openings of the mesh by changing the percentage of acrylamide in the reaction. So the mesh nature or the sizes of the mesh, the spaces in them can be adjusted. Higher percentages of acrylamide give smaller openings and are more effective for separating smaller molecules, whereas lower percentages of acrylamide are used when resolving mixtures of larger molecules. So larger percentage for small molecules, lower percentage of acrylamide for larger molecules. Now note, polyacrylamide gels are also used to separate small nucleic acid fragments, with some acrylamide gels capable of separating pieces of DNA that differ in length by just one nucleotide. So although you are thinking more of proteins, you can still use this for nucleic acid separation. And then the information is saying that depending on how you do it, you can even use it to separate as small as one nucleotide length difference in nucleic acids. Now the next alteration that can be done is charge alteration by SDS. Now a second consideration is that proteins must be physically altered to present themselves to the matrix like the negatively charged rods of DNA. You remember we said the DNA phosphate backbone has same charge, negative charge. So you try to do same to the proteins. This is accomplished by treating the proteins with an ionic detergent SDS, that's sodium dodecyl sulfate. Now the SDS denatures the proteins so they assume a rod-like shape. We mentioned that proteins is globular, round, but now we are pushing them into rod. So the SDS denatures the proteins so they assume a rod-like shape, and the SDS molecules coat the proteins such that the exterior surface is loaded with negative charges. So you coat the outermost to load it with negative charges, masking the original charges on the proteins and making the charge on the proteins more proportional to their mass, like the backbone of DNA. So you are finding a way of pushing the proteins to behave similar to the DNA by making them negatively charged, and then the extent or the size would increase the number of negative charges or decrease the number of negative charges depending on the size. Bigger, more negative charges; smaller, fewer negative charges. Now, since proteins typically have disulfide bonds that prevent them from completely unfolding in detergent, samples are boiled with mercaptoethanol to break the disulfide bonds and ensure the proteins are as rod-like as possible in the SDS. So another way of causing the proteins to behave in the manner you want is to have them boiled with mercaptoethanol. And this mercaptoethanol is supposed to break disulfide bonds. Mercaptoethanol breaks disulfide bonds and then makes the proteins more rod-like, like your nucleic acids. Now a third consideration, that is stacking gel. A third consideration is that a stacking gel may be employed at the top of a polyacrylamide gel to provide a way of compressing the samples into a tight band before they enter the main polyacrylamide gel. So you are finding a way of packing your content before you introduce them to the separating gel. So at the top where you are going to load your samples, you employ a stacking gel. And this provides a way of compressing the samples into a tight band before they enter the main polyacrylamide gel, that is the resolving gel. Now, just like DNA fragments in agarose gel electrophoresis get sorted on the basis of size, the proteins migrate through the gel matrix at velocities inversely related to their size, so larger, slower, smaller, faster. Upon completion of the electrophoresis, proteins may be visualized by staining with compounds that bind to proteins like Kumasi brilliant blue or silver nitrate. Now, mind you, the Kumasi is not your Asante Kumasi. It's COOMASSIE, brilliant blue, or your silver nitrate. So, use of a stacking gel can also help make the use of the gel separation for proteins much better. So, now non-denaturing gel electrophoresis. The SDSP technique described above is the commonest method used for electrophoretic separation of proteins. In some situations, however, proteins may be resolved on so-called native gels in the absence of your SDS. So your SDS denatures, but now we are using native gels. Under these conditions, the movement of proteins through the gel will be affected not simply by their mass, but by their charge at the pH of the gel as well. Proteins complex with other molecules may move as single entity, allowing the isolation of the binding partners of proteins of interest. So under the non-denaturing gel, we are now using other molecules to cause the movement of the proteins, unlike in the SDS where we denature them. Then we have the isoelectric focusing. Now, you know that proteins have different amino acids. Amino acids have different charges, and the charges that they have causes them to have pH, varying pH. So proteins vary considerably in their charges and consequently in their pI values. That's pH at which their charge is 0 is the pI value. This can be exploited to separate proteins in a mixture because they are varying pIs or pHs. Separating proteins by isoelectric focusing requires establishment of a pH gradient in a tube containing an acrylamide gel matrix. So in this one, the pH across the gel varies. So it's a tube, but the pH varies in gradients. The pore size of the gel is adjusted to be large to reduce the effect of sieving based on size. So size is not our consideration. So we give it enough space for everybody to move. Molecules to be separated are applied to the gel containing the pH gradients. An electric field is applied. Under these conditions, proteins will move according to their charge. Proteins will move according to their charge. So there is a gradient and each person is moving based on its charge because we've increased the size of the pores of the gel so that the size, movement by size, is not the major factor at this point. Now, positively charged molecules, for example, move toward the negative electrode, but since they are traveling through a pH gradient, as they pass through it, they reach a region where the charge is 0, and at that point, they stop moving. They stop moving because they have no charge, their charge is 0. They are at that point attracted to neither the positive nor the negative electrode and are thus focused, isoelectric focusing at their pI. Using isoelectric focusing, it is possible to separate proteins whose pI values differ by as little as 0.01 units. So in using the isoelectric focusing, proteins are moving based on their pH, or better still, their pI. At the point where their pH equals the pH of the region they've reached, their charge is 0. Then they get stuck. So every protein will get stuck at the point where it has a 0 charge. And that is how isoelectric focusing is used. Now we have 2D gel electrophoresis. 2D gel electrophoresis. Both SDS page and isoelectric focusing are powerful techniques. Individually, they are powerful, but a clever combination of the two is a powerful tool of proteomics. You know, proteomics is the science of studying all of the proteins of a cell or tissue simultaneously. Now, in 2D gel electrophoresis, a lysate, when you crush all the cells, you have a lysate. A lysate is first prepared from the cells of interest. The proteins in the lysate are separated first by their PI through isoelectric focusing. Because you know isoelectric focusing will push them into their PIs or will separate them based on PI. Then you use SDS page to separate them by size. And this is how it is. So in this picture, your first is to introduce your lysate into the isoelectric focusing. Then into the isoelectric focusing separates them based on their PI or charge. And then at 3, you turn it 90 degrees. Then the SDS will now separate those at a particular charge into their different forms based on size. So you first separate them based on charges, you turn them 90 degrees, then you separate them based on size using the SDS page. So the 2D gel electrophoresis uses first isoelectric focusing and then second SDS page separation. So these are the steps. Step 1, mixture of proteins is first applied to a tube or strip where isoelectric focusing is performed to separate the proteins by their PI. Then the next step, as shown in the picture above, the gel containing the proteins separated by their PIs is turned on its side and applied along the top of the polyacrylamide slab for SDS page to separate on the basis of size. Then step 3, the proteins in the isoelectric focusing matrix are electrophoresed into the polyacrylamide gel and separated on the basis of size. That's what I explained earlier. Now the product of this analysis is a 2D gel as shown in the figure. The power of the 2D gel electrophoresis is that virtually every protein in a cell can be separated and appear on the gel as a spot defined by its unique size and PI. In the figure, spots in the upper left correspond to large positively charged proteins, whereas those in the lower right are small negatively charged ones. So upper left and then lower right. Every spot on a 2D gel can be eluted, so you can pick each spot and identify it by using high throughput mass spectrometry. This is particularly powerful when one compares protein profiles between different tissues or between control and treated samples of the same tissue. So using the 2D gel electrophoresis, you can separate the proteins in a cell into its individual contents or proteins. Now, protein profiles can be compared based on this. So comparison of 2D gels of proteins from non-cancerous tissue and proteins from a cancerous tissue of the same type provides a quick identification of proteins whose level of expression differs between the two. Information such as this can be useful in designing treatments or in understanding the mechanisms by which cancer develops. So if you do a 2D gel of proteins from a cancerous cell and a non-cancerous cell, you can compare and know where the changes are and then zoom in and deal with them. That's how powerful molecular biology can be in diagnosis. Now we come to utilization of molecular biology. So we're looking at DNA chips, automation, gene therapy, then applications in diagnosis of genetic disorders. We talk briefly about the Human Genome Project and then ethical considerations. So what are DNA chips? Microarrays can be used to study the extent to which certain genes are turned on or off in cells and tissues. In this case, instead of isolating DNA from the samples, RNA, which is a transcript of the DNA, is isolated and measured. So in this case, you are measuring the RNA. Today, DNA microarrays are used in clinical diagnostic tests for some diseases. Sometimes they are also used to determine which drugs might be best prescribed for particular individuals, because genes determine how our bodies handle the chemistry related to those drugs. So depending on how your genes are, particular drugs would apply to your body in a different way. With the advent of new DNA sequencing technologies, some of the tests for which microarrays were used in the past now use DNA sequencing instead. But microarray tests still tend to be less expensive than sequencing, so they may be used for very large studies as well as for some clinical tests. So in microarrays, you have a solid phase onto which you'd apply your sample. If what you are looking for is available, it binds to a particular template, and then that template can be identified to let you know that what you are looking for is embedded onto your solid phase. So to determine whether an individual possesses a mutation for a particular disease, the scientist first obtains a sample of DNA from the patient's blood, as well as a control sample, one that does not contain a mutation in the gene of interest. The researcher then denatures the DNA in the samples. That's a process that separates the two complementary strands of DNA into single-stranded molecules. Then the next step is to cut the long strands of DNA into smaller, more manageable fragments, and then to label each fragment by attaching a fluorescent dye. Now there are other ways to do this, but this is one common method using the fluorescent dye. You can radio type them, etc. The individual DNA is labeled with green dye, and the control or normal is labeled with red dye. Both sets of labeled DNA are then inserted into the chip and allowed to hybridize or bind to the synthetic DNA on the chip. That's a solid phase. If the individual does not have a mutation for the gene, both the red and the green samples will bind to the sequences on the chip that represent the sequence without the mutation. So there's a sequence without the mutation to which these single strands that you are introducing, or the fragments you are introducing, can bind to. If what you are bringing is, or better said, the mutated one, the gene, and then the control are all not having any mutation, then you realize that all of these will successfully bind to the sequences on the chip. However, if the individual does possess a mutation or has a mutation, the individual's DNA will not bind properly to the DNA sequences on the chip that represent the normal sequence, but instead will bind to the sequence on the chip that represents the mutated DNA. So on the chip you have both the normal and the abnormal. So when you introduce and you are not able to bind to the normal, if you have a mutation, then you bind to the indicator for the mutation. That's the DNA chips. Gene therapy. Now, gene therapy is a medical approach that treats or prevents disease by correcting the underlying genetic problem. So there's a disease, there's an underlying genetic challenge that is causing that. So in gene therapy, you look out for that and treat based on that. Gene therapy techniques allow doctors to treat a disorder by altering a person's genetic makeup instead of using drugs or surgery. The earliest method of gene therapy, often called gene transfer or gene addition, was developed to introduce a new gene into cells to help fight a disease, or introduce a non-faulty copy of a gene to stand in for the altered copy causing the disease. So you have a gene that is a mutated gene that is causing the disease. In gene therapy, scientists also would rather introduce a positive gene or a gene that would help in the situation, and that will be introduced into your genome to help fight or correct the error. Now there's a newer technique called genome editing, which uses a different approach to correct genetic differences. Instead of introducing new genetic material into cells, genome editing introduces molecular tools to change the existing DNA in the cell. So this one you are not introducing, but you are correcting the DNA that is already there. And genome editing is being studied or processed in the following. You fix a genetic alteration underlying a disorder so the gene can function properly, or you turn on a gene to help fight a disease, or turn off a gene that is functioning improperly, or remove a piece of DNA that is impairing gene function and causing disease. So in gene editing, you can use any of these options to do your editing. Fix a gene alteration that is underlying a disorder so that the gene will function properly, or you turn on the gene, or turn off a gene, or remove a piece of DNA that is impairing gene function. Now, there are various types of gene therapies based on the cell type. So basically, there are two types: the somatic and then the germline. The somatic gene therapy and then the germline gene therapy. So for the somatic gene therapy, you transfer a section of DNA to any cell of the body that doesn't produce sperm or eggs. Somatic body cells. You know, somatic is body cells. Effects of gene therapy will not be passed on to the patient's children. So when you do the somatic gene therapy, that is dealing with any non-sperm or egg genes, the effect of it will not be passed to its children. And then the next one is the germline gene therapy. Now you transfer of a section of DNA to cell that produce eggs or sperm. Effect of gene therapy will be passed on to the patient's children and subsequent generation because you have touched the reproductive cells. The reproductive cells. Now there are various gene therapy techniques, and these include the gene augmentation therapy, gene inhibition therapy. Now in gene augmentation therapy, this is used to treat diseases caused by a mutation that stops a gene from producing a functioning product such as a protein. So if there's a mutation that prevents the production of a particular protein, then gene augmentation would be useful. And then gene inhibition therapy. Now in gene inhibition therapy, this is suitable for the treatment of infectious disease, cancer, and inherited disease caused by inappropriate gene activity. So if there's an inappropriate gene activity, then a gene inhibition therapy would be useful. The aim of this is to introduce a gene whose product either inhibits the expression of another gene or interferes with the activity of the product of another gene. So you are doing an activity or a gene therapy which will inhibit the expression of another gene. So in the presence of that gene, the other one doesn't show, or better still, interferes with the activity of the products of the another gene. So the gene is allowed to produce what it wants to produce, but then you now interfere with the products of that gene. Now you can also kill specific cells. Now this is suitable for diseases such as cancer. that can be treated by destroying certain groups of cells. So in this one, your agenda is to kill particular cells. Now the aim is to insert DNA into a disease cell that causes that cell to die. So you introduce a DNA into a disease cell, and then that will cause that cell to die. Now there are challenges of gene therapy. Now delivering the gene to the right place and switching it on is a challenge. Now avoiding the immune response, because if you are introducing something that is not there, it can initiate an immune response. So avoiding the immune response. Then making sure the new gene doesn't disrupt the function of other genes. So you brought in a new gene, you make sure it doesn't disrupt the function of other genes. And then also the cost of gene therapy. Now let's come to the HGP, the Human Genome Project. Now the Human Genome Project was one of the great feats of exploration in history. Rather than an outward exploration of the planet or the cosmos, the HGP was an inward voyage of discovery led by an international team of researchers looking to sequence and map all of the genes, together known as the genome, of the members of our species, that's humans, Homo sapiens. Now the whole thing began in October 1st, 1990, and was completed in April 2003. So that's about 13 good years. The Human Genome Project gave us the ability for the first time to read nature's complete genetic blueprint for building a human being. So based on the Human Genome Project, now we know all the genes that make up the human being. Now the Human Genome Project has revealed that there are probably about 20,500 human genes. This ultimate product of the Human Genome Project has given the world a resource of detailed information about the structure, organization, and functions of the complete set of human genes. This information can be thought of as the basic set of inheritable instructions for the development of a human being. The full sequence was completed and published in April 2003. So after the Human Genome Project, we know there are 20,500 human genes, and it gives us the plethora or the total set of genes that can be inherited from one person to the other. Now, there are ethical considerations based on the Human Genome Project. However, the momentous implications for individuals and society for possessing the detailed genetic information made possible by the Human Genome Project were recognized from the outset. So now you know that there are implications for knowing the full human genome. Another consideration or major component of the Human Genome Project and an ongoing component of the NHGRI is therefore devoted to the analysis of the ethical, legal, and societal implications. So out of the Human Genome Project, we have the NHGRI, which is now devoted to the analysis of the ethical, legal, and social implications of our newfound knowledge and the consequent development of policy options for public consideration. Now, having known the human genome, there is the need to have restrictions on how it is applied. So when I go into a company and I'm being employed, can they check my genome and know that I do not possess this particular gene, so I cannot work with them? These are considerations that need to be looked at, especially now that we know the full genome of humans on the earth. Thank you.